drug is sometimes zero because of this process. So just to remember briefly, okay, the cells of the nervous system are the neurons and the glial cells. Okay, we are not going too deep into this because when we study nervous system itself, we are gonna go in more detail. Okay, the most important cell of the nervous system is the neuron. It's the functional unit of the nervous system. Okay, there are 10 to the 10 neurons in our body. Notice that I say body and not brain or nervous system, because there are many of these neurons in the gastrointestinal tract. Okay, in the gastrointestinal tract, we have a part of the nervous system that is called the enteric nervous system that contains as many neurons as the spinal cord. Okay, so many neurons around the body. The neurons are connected to each other. They receive information from different neurons, from thousands of other neurons, allowing to process information, integrating that information, and determining which is the best motor action or effect, okay, that is that we need to perform at every moment. Okay, this participates in many conscious and unconscious processes, emotions, learning, memory, and different other processes that happen in our nervous system. We are going to see how they receive information from different sources, how they integrate that information, and then they transmit this either to other neurons or to effectors that could be muscles, could be glands. Okay, and these neurons are classified into afferent when they receive information, efferent when they send information to effectors, and interneurons which integrate the signals and they take the decisions for us, sometimes conscious or sometimes unconscious. Okay, as you know that many of the functions in the body, most of the functions are carried at the unconscious level. So when they affect the muscles, glands, using neurotransmitters mainly. Now, take a look at this diagram. Okay, here I've tried to make a representation of a neuron, okay, that is receiving information from a couple of other neurons. Notice that one neuron, the one on top, okay, is making a synapses at the level of the dendrite. The other neuron here is making a synapses at the level of the body of this neuron, at the level of the soma. Okay, then there is an axon from this neuron that will go to either another neuron or effectors, muscles, glands, etc. But in the middle of this axon, we can see the axon of a third neuron that is making a contact there, another synapses. This gives you an idea of the way neurons communicate with each other and participate in the decision-making process that happens within our brain, spinal cord, and also gastrointestinal tract. Okay, you have here the classification of these synapses. For example, this type of synapses up here is called axodendritic synapse. Okay, this neuron is receiving information from another neuron through one of its dendrites. Dendrites, there can be many per every neuron. They normally receive signals. Okay, and on the left side, we have an axosomatic synapse. Okay, it's a synapse that is occurring in the body, soma of the neuron. This is important, for example, because this type of synapse is the one that neurons need for stimulation to keep them working. Okay, this is the synapse that, for example, gives growth factors to the neurons to maintain them healthy and strong. And then we have the axonic synapse on the right lower part. Okay, that regulates, this type of synapse here is the one that neurons use to regulate the neuronal function. Okay, for example, let's say this neuron fires to stimulate another structure that is here. Maybe this axonic synapse releases a neurotransmitter that increases the nerve transmission here, okay, to reinforce what this neuron wants to do. But in some cases, this other neuron will release here inhibitory neurotransmitters to stop this neuron from firing. Okay, this type of connection here, the axonic, is the one that the nervous system uses to modulate the nerve transmission. Modulation means either increasing or decreasing the propagation of the action potential, okay, to either reinforce or to inhibit what is going on or the process that is going on here. Now, there is a part here that we haven't mentioned yet, that is the axon hillock. Okay, the axon hillock, or the initial segment of the axon, is the area of the neuron that integrates and summates all the input that the neurons are receiving. Here I just represented one axodendritic synapse, but there can be hundreds of these axodendritic synapses, okay, producing different signals, releasing different neurotransmitters. Okay, so the hillock will integrate and summate all of these impulses that the neuron is receiving, and depending on the summation that is occurring in the hillock, an action potential will either occur or not. So now let's move to the left part. Okay, neurons have sensors that are these dendrites that receive the input from many different other neurons. Okay, that are, and these impulses will be transmitted here to the soma. Okay, so some of these impulses, maybe if they are far away from the axon hillock, may be lost. If they are closer to the axon hillock, may reach the hillock. Okay, but in many cases, all of the processes that occur in the axon, in the soma, as a result of all of this stimulation or inhibition from many different neurons, is going to reach the axon hillock, and the hillock is going to take the decision. The hillock is the integration or control center of the neuron, the one that decides, okay, if an action potential will take place or not. And the axon itself, okay, is the output or effector of the neuron, okay, after carrying all of this action potential to the axonal end, okay, one effect will occur. Okay, the axon end will release some neurotransmitters that will either stimulate or inhibit the following neuron. Okay, that occurs at the level of the axon terminal, and is a process that we are going to be studying, okay, when we see the part of synapses. Now, I have here an analogy, okay, that will help you understand this better. So I'm gonna take something that I made, okay, to try to make students understand this better. Let me see, let me know please if you see the new slide that I have here.
We can see it.
Thank you so much. Uh-oh. One second. Okay. So to understand this, I always like to use an analogy from real life. Let's say we go to a restaurant. Okay, we are deciding if eating in a restaurant or not. Okay, we before taking that decision, we need to analyze different things about that restaurant. Okay, we enter into the place and we notice the smell of the food. All of this information enters through the nose and goes to the brain cortex. Maybe in some places they give us a sample. Okay, we taste the food, or we imagine that it tastes well. We look at the place. Is it a nice place? Do I like the decoration? Is there nice music, or is there a very loud music? How is the temperature of the place? Is it very hot? Is it very cold? We take a look at the menu. Can we afford it? Do we like the price of the food? Do we like the price of the wine? Is there nice people around? Or is there a lot of people making noises and fighting? And do we like the different things that are in the menu? Okay, notice, and I just made 8 things in this list, but we actually look at many other things. Okay, all of this information is going to enter to different areas of the cortex, and it's going to stimulate certain neurons there, and all of these neurons are going to fire certain information to areas of the brain that we use for decision making. Okay, so if we give a value of 1 point to each of these things, okay, and we imagine that all of these things are going to affect a single neuron that could be this one, okay, we can understand that, for example, this neuron is going to be analyzing and adding or subtracting different points before taking a decision. So let's say we like the smell, the taste, the sight, the sound, the temperature, but we don't like the price and we don't like the people, and the menu is not very interesting to us. Okay, the first 5 components are gonna stimulate the neuron, while the last 3 are gonna inhibit the neuron. But maybe we take the decision of staying in that place to eat. But let's say we don't like the temperature. Okay, now it's half and half. Probably we are not gonna stay in that place. Maybe we are gonna look for another place. And if we even don't like the smell of the place, or I don't like, this smells like rotten food, of course, we are not gonna stay in that place. Okay, so control centers, the axon hillock of the neuron, will take all of the information that is receiving from different neurons, and it's gonna add and subtract the factors. And if the factors, the stimulating factors, are above the inhibitory factors, an action potential will occur. If there are more inhibitory than excitatory factors, okay, then an action potential will not take place. Okay, it's yes or no, all or none response. Okay, that I'm sure you studied in the past. That is what characterizes the action potential. So here we are going to study that neurons actually have different types of membrane potential. Okay, there is one membrane potential that is called the resting membrane potential. That is the one that we could say is the baseline potential of the cell when the cell is not making anything. And then we have potentials that occur here in the sensors that can be excitatory or inhibitory. These are called receptor potentials or graded potentials, sometimes known as local potentials, because they occur only here at the level of the dendrites. That can be excitatory or inhibitory. I like, I like, I like, I like, I don't like, don't like, don't like, and don't like. But the action potential, okay, the one that occurs at the level of the axon, is an all or none phenomenon, okay, that will occur depending on the decision-making process of the hillock. When the action potential reaches the axon terminal, something will happen here. This electricity will make some channels open at the level of the axon terminal. Okay, calcium, for example, enters here, and this axon terminal releases neurotransmitters that will either stimulate or inhibit the following neuron. And in this other neuron, the same thing that is happening here will happen, because I am representing the connection between this neuron and this neuron, but this one is also receiving stimulation from many others. So this other neuron will determine if the information will continue or not. And that will happen in our body. Okay, for example, every time we are going to do any kind of movement, if someone tells us, okay, I need you to bring me water, okay, that information will enter through my ears, will activate the brain cortex, the auditory cortex. Then I need to remember where the water is, or in the refrigerator. I need to look using my eyes to the refrigerator, to the floor. I need to walk there. Then I need to open, I need to take the water, put it in a cup, and then move back using my vision, my ears, my muscles, and give the water to this person. So just imagine how many neurons are transmitting to each other, how many decisions are being taken at the same time, just to perform a simple action like giving water to someone. Okay, so let's move back to our presentation after seeing this, because it's important to understand the differences between the several types of membrane potential that occur in our brain, in our nerves. Okay, just to remember what these structures are. The cell body or soma is the one that has the nucleus, okay, different organelles, endoplasmic reticulum, smooth and rough. Okay, the dendrites are the afferent or receptive part of the neuron. Okay, there are different number of dendrites depending on the type of neuron. Okay, depending on if they are unipolar, bipolar, multipolar. The axon is the efferent or transmission zone of the neuron. Is one of the most important structures in signal transmission. Axons can be very, very, very long. Just imagine a guy who plays basketball, who is a very tall guy, 7 feet tall. And imagine how long the axons of the neurons, of the nerves of his legs are. Imagine they start at the level of the spinal cord, and the axon extends all the way down to the feet. Okay, some axons are very short, some axons are extremely long. Okay, it contains the initial segment, which is the hillock, and then the axon, the proper extension, and the terminal synaptic button or axon terminal. And there is a transport of neurotransmitters that is very interesting to when you learn about it. Okay, when I started studying biology and medicine, I thought that neurotransmitters were made there in the soma, okay, in the endoplasmic reticulum, and they simply travel down the axon by diffusion. They were like pushed, I don't know, by internal currents of the cells. But then I learned that that is not the way they travel, that they actually are carried by proteins, okay, along microtubules that are within the axon. Okay, and then these neurotransmitters would travel from the soma of the neuron towards the end of the neuron by a type of transport that is called antegrade or anterograde. You can use any of the two words. And then some of these neurotransmitters would be taken back for recycling in a type of transport that is known as retrograde. And that is a very, very interesting. There is a video on YouTube that actually shows how this works. And the way these proteins transport the neurotransmitter is amazing. You can see them like walking along these microtubules. And then we have the synapses. The synapses are the structure that allows the communication between different neurons, or neurons and glands or muscles. And as you saw in the diagram before, these synapses may occur between axons and dendrites, axons, and the axons and the soma. And then we have the neuromuscular junction. The neuromuscular junction is another type of synapse, but in this case, is between a neuron and the muscles. And we enter here into the part of membrane potentials. Before moving there, is there any question? Is there any question, please? I don't want to move on if anyone has any kind of question. If you have questions and you don't want to tell using your voice, you can simply write it in the chat. And maybe during the break, I can see it and go back to this. So let's take a look at the membrane potentials. We were telling this before, there are basically three types of membrane potentials. Okay, compare the word potential, remember too, when we have different types of batteries, 1.5, 9 volts, etc. So different cells will have a different membrane potential, higher or lower capacity to do work. Okay, when I say work, I'm telling about the capacity to transmit signals. A membrane potential, or a difference in electrical voltage, which is the definition of a potential, okay, is the difference in charges, electrical charges, between the interior and exterior of a cell membrane. If we could place an electrode inside a cell, okay, and we would measure the voltage inside the cell, we would see that there is a difference between the outside and the inside of the cells. Okay, generally, the interior of the cell is negative compared to the exterior of the cells. Okay, there are different chemical, electrical properties and physical properties that influence that. Okay, but what we need to know is that there are more negative charges inside a cell when the cell is at rest, okay, than outside the cells. And the basics for this is that the cell has more permeability for potassium than for sodium. Okay, that means there is more potassium leaving the cell than sodium entering the cell. Okay, cell membranes have more potassium channels than sodium channels. And when potassium leaves the cells, the anion that belongs to that potassium cannot go out and stays inside the cell, leaving a more negative environment inside the cell than outside the cells. Okay, that is the basis for the generation of the resting membrane potential, and that's why it's always negative, let's say minus 50 millivolts, minus 60, minus 70, minus 90 millivolts, depending on what type of cells we are talking about. And then we have the graded potentials or local graded potentials, also known as receptor potential. Okay, the one that occurs in the dendrites, remember, can be excitatory or inhibitory. Okay, this is a potential, and it's important to understand and have very clear the differences between these local graded potentials and the propagated action potentials. Okay, the graded potentials vary in amplitude. Okay, can be larger, okay, or smaller. They can be summated. They can have spatial and temporal summation. We are going to see what that means in a while. This can be excitatory or inhibitory. Okay, when they are excitatory, okay, we can use another word for excitatory and can be depolarizing. We're going to see why. We're going to see examples in a little bit. Or can be inhibitory or hyperpolarizing. And after the summation of all of these excitatory and inhibitory potentials, okay, they may produce an action potential if the threshold for the action potential is reached and exceeded in the action hillock or action initial segment. There are other terms for these local graded potentials. Okay, I told you before receptor potential, but another term for this is postsynaptic potential. Okay, because it's the one, if we go back here, is the one that occurs after the synapses. Okay, this neuron on top here fires, releases a neurotransmitter. Neurotransmitter can be either excitatory or inhibitory, can be depolarizing or hyperpolarizing, and it will produce changes in this dendrite that is postsynaptic, okay, compared to this one here that is the presynaptic. That's why this is also known as postsynaptic potential. And then we have the classic action potential that for sure we have studied many times, that is called propagated action potential. Okay, this consists of different cycles of depolarization, repolarization, hyperpolarization, and going back to the resting membrane. Okay, this may last for 1 to 2 milliseconds. It's very fast. Okay, and it's generated by voltage-gated channels, normally cation channels, which are sodium and potassium channels. Okay, the local graded potentials can be generated by, for example, other types of channels that we classify as ligand-gated channels. Let me put that here. Ligand-gated channels. What is a ligand? A chemical that can be a hormone, can be a neurotransmitter, okay, that binds to the receptors and opens or closes them. For example, epinephrine may bind to a receptor, acetylcholine may bind to a receptor and stimulate it to open, and this receptor is generally a sodium channel, so sodium will rush into the cells. Okay, but it's a chemical like acetylcholine, a ligand, but it's opening the local or these dendritic receptors, producing a local graded potential. In the case of the axon, it's not acetylcholine what binds to the axon channels. It's simply when there is a change in the voltage in that area, okay, the sodium channels will open or close. That's why they are called voltage-gated channels versus ligand-gated channels in the case of the dendritic channels or the postsynaptic channels. And here we have a classic representation of how we measure, okay, the potentials in the cells. Okay, in the center we have the resting potential, resting membrane potential. Notice that here we have two electrodes. One electrode is inside the cell, in the cytoplasm, okay, and this will measure the difference between the inner part of the cell and the outer part of the cell, which is the control. Okay, there is a difference in electrical potential or voltage of minus 80 millivolts, negative 80 millivolts. Why the cells have this difference in potential? Because of the action of the sodium-potassium pump. We were talking during the last encounter about steady state. Okay, we were explaining that the cells inside have a lot of potassium compared to the exterior. In the exterior, there is a lot of sodium compared to little sodium inside the cells. And we were explaining a bit ago that there is a lot of more potassium leaving the cell by leaky channels that are always open than sodium entering the cell. But the anions can't leave, so there are more negative charges. The anions that normally were paired with this potassium that is moving out, okay, so negative charges predominate in the inner side of the membrane, okay, due to the action of the sodium-potassium pump. If the sodium-potassium pump wasn't working, then sodium would rush in, potassium would go out, and there would be an equilibrium, and there would be no difference in electric potential, and that means death. Now, what happens, okay, if, for example, we open a sodium channel in this membrane? Since there is a difference in the concentration of sodium, there is a lot of sodium outside, a lot more than inside, and there are negative charges inside the cells. If you open a sodium channel, sodium will move in because there are two forces driving it inside the cell. Sodium is positive and there are negative charges inside the cell. So there is an electrical gradient. And also sodium has a lot more concentration outside than inside. So there is a chemical gradient difference in concentration. So this electrochemical gradient will drive sodium inside. So we are going to add lots of positive charges to the inside of the cell, and notice how the membrane potential becomes positive. Okay, now there are more positive charges inside than outside. There is a positive 30 millivolts difference in potential. Now, we are not going to stay like this forever. Okay, when the inner side of the cell reaches this value, okay, there are some voltage gated channels that will open when we achieve this value of potential, of voltage. These are potassium channels. Okay, potassium channels will open and potassium is going to start moving out. Lots of potassium moving out, moving out, moving out, moving out. Potassium has a positive charge. Okay, and this will lead to the inner side of the cell reaching a very negative value compared to the initial one. Okay, when this membrane potential is going from the positive value to the negative value, we call that repolarization. And when we reach a value that is a lot more negative than the resting membrane potential, we call that hyperpolarization. And then everything is gonna go back to normal because the sodium-potassium pump is gonna reestablish the initial steady state. Okay, this is in a nutshell what happens. Okay, what explains the resting membrane potential, what explains the depolarization, and what explains the hyperpolarization that occurs in these membranes. So I was explaining before that there are different types of potentials. Okay, some of them are graded, local. Some of them are, or one of them is all or none. Okay, action potential. And I was saying that there is a process called summation, that some of these potentials may be depolarizing or excitatory. Some of them may be hyperpolarizing or inhibitory. Okay, and that the summation of all of these depolarizing and hyperpolarizing potentials, okay, is what lead to the axon hillock to either reach or not reach the threshold. And that by reaching the threshold of the neuron, okay, is the mechanism by which these neurons will either produce fire and action potential or not. Okay, we are going to review that in a little bit, and we're going to go over these concepts again, so I make sure that you have all of these very clear. Okay, we are going to be having a break, okay, 10 minutes break. 8:50, almost 8:50, so let's meet again at 9. If there is any question before going to the break, I will gladly answer. If not, just simply write it in the chat and we go over those things when we come back from the break. Is there any question? No questions? Okay. Okay, see you in 10 minutes, okay? Okay, so we are ready to enter, okay, in the second part of this really complex topic of the electricity in the cells. Okay, there are many things when you watch the videos that I recommended there. You are going to learn certain things about electricity. And there are other videos. If you want to do it, you can explore these videos that compares how the cell membranes work in a similar fashion. Okay, that different devices that we have, electrical devices. Okay, if you want, you can go and study and look online for the word capacitance. And capacitor, okay, and see how the cell membrane is like a capacitor. If you know something about electricity and electrical circuits, you may know what a capacitor is. And if you want to get amazed about these things, look for cell membrane as a capacitor, and you're going to learn, okay, how this works for real and how it can be compared with certain devices that we use every day. Okay, and being amazed about the human body is one of the things that everybody gets after studying all these things. So let's take a look at this. Okay, here we have a representation, okay, of different potentials that may occur in a cell membrane. Okay, if you focus here on -70, we could say that this is the baseline or resting membrane potential of a cell. This is going to be different, okay, for different cells, muscle, neurons, etc., smooth muscle, skeletal muscle, cardiac muscle. They have a different resting membrane potential, but we're going to focus now on the neurons, okay, to see how they integrate all of the different stimuli that they are receiving. Okay, and what is, yes?
Your screen.
I'm not sharing?
No.
Oh, thank you, thank you. Thank you so much. Okay, now we are. So I was saying, we are going to focus here on this part, okay, on the different membrane potentials that you see here. The -70 would represent the baseline or resting membrane potential. And these are different stimuli that a neuron might be receiving. Okay, and this will help to understand the difference between a local graded potential and an action potential. Okay, here we have a stimulus that is small in magnitude. Let's say acetylcholine binds to some receptors in the dendrite, some sodium channels open, some little bit of sodium goes inside the cell, making the membrane a little bit less negative, but just a tiny bit. Okay, there can be a stronger stimulus opening a bit more sodium channels, so more sodium comes inside the cell. And notice that it only reached -60. Okay, we need that the inner part of the cell, more exactly at the axon hillock, reaches this line here, the -55, which is the threshold. Okay, this threshold is the value, the voltage at which the axon hillock will initiate an action potential. Because in the hillock, there are some sodium channels that respond to voltage. That's why they are called voltage-gated sodium channels. Okay, the third stimulus is a stimulus that has a longer duration, but not too much magnitude, not too much intensity. It kept the sodium channels open for a longer time, but it's not enough to reach the threshold. And here we have a larger stimulus. Okay, we have a lot more sodium channels open, so there is a lot of sodium entering and reaching the axon hillock in a way that the inner part of the axon hillock reaches threshold. At that point, sodium channels in the axon hillock will open and an action potential will take place. Okay, and then we are going to have a series of sodium channels along the axon that, again, they respond to voltage, and that will permit the transmission of this electrical impulse along the axon. All of these stimuli that we saw here on the left part of this slide are producing a depolarization. So the inner part of the cell becomes less negative. That's why they are called depolarizing graded potentials. Okay, we could say, instead of depolarizing, we could say excitatory. So we apply this more to our topic, excitatory potentials. Okay, and in biology and in physiology or medical clinical physiology, we call this type of potentials excitatory postsynaptic potentials, EPSP. If we move to the right side here, we are going to see other types of potentials that instead of depolarizing are producing a hyperpolarization of the membrane. Okay, so all of these, you're going to see that the distance, okay, goes a lot more farther away from the threshold. So it's going to be a lot more difficult to reach the threshold. And you have to imagine that, for example, a neuron is receiving two potentials at the same time. Let's imagine that a neuron is receiving this excitatory potential that normally makes it reach the threshold, but at the same time is receiving from other two dendrites this hyperpolarizing and this hyperpolarizing. When you summate the action of this stimulus, stimulus 1, and you subtract the effect of a stimulus 2 and a stimulus 3, you're going to see that, okay, the result of all of these, this minus this minus this, is going to be a value that is not going to reach threshold. Let's say around here. So even though we are trying to stimulate the neuron using this stimulus, these other two stimuli are going to inhibit the neuron and it's not going to fire. If we go back to our example in the restaurant, let's say that we love the menu. Oh my goodness, it's a great menu. I love the food here and the price is amazing. But there is a very loud music and it is very cold. So even though I like the menu and the price, I don't want to stay here because this is a very loud place and very cold, and I really don't want to have dinner in a place that is so cold and with so loud music. So no dinner here, let's look for another place. Okay, so hyperpolarizing graded potentials are the ones that inhibit the neuron. That's why we also call them inhibitory postsynaptic potentials or IPSPs. Now, sometimes our nervous system does different things in order to transmit signals. Okay, we were talking about different excitatory and inhibitory potentials. Okay, sometimes if you want to stimulate a neuron, okay, you can do different things. Either produce one excitatory potential that is very high in magnitude, or produce many different small potentials that when you add them all, you're going to also reach the threshold. Why that happens? Because let's say this is the soma of a neuron. You are producing a stimulus here, sorry, another here, another here, and another here. The sodiums, all of the sodium ions that enter inside the cell through different channels is gonna by diffusion travel, okay, in the inside of the neuron and reach the hillock. So if we add all of these stimuli, we may reach the threshold at the level of the hillock, and that is what is important. Okay, so let's take a look at the types of summation. One of them is called spatial summation. Okay, notice that here we have different neurons that are stimulating a single neuron. Maybe each of these stimuli is not enough to reach threshold, but when you add them all, okay, when you add all of these excitatory postsynaptic potentials, the effect at the level of the hillock is going to be an action potential. Okay, we summate the different stimuli that the neuron is receiving through different dendrites. Simultaneous summation, simultaneous stimulation by several neurons. When we add them all, we may reach a threshold and produce action potentials. Another way is simply the temporal summation. In this case, we have a single neuron that is trying to stimulate another one. Okay, in this case, the single neuron is gonna produce several excitatory potentials, but with a very short distance between them, with a very high frequency. Compare the frequency of the excitatory potentials here, the frequency, with the frequency in the one above. Let's say on top, on the one above, we have 4 stimuli per second, and the one below, we have 10 stimuli per second. That is temporal summation, high frequency stimulation by one presynaptic neuron. And at the end, the effect will be the same. Okay, we send this stimuli very fast, sodium enters very fast inside the cell, and there is no time for sodium to move out, so the axon hillock reaches threshold, and an action potential is produced. Okay, here we have 3 examples on the right side. On the first one, we have excitatory and inhibitory postsynaptic potential cancellation, because we have an excitatory and an inhibitory stimulus of the same magnitude, so the effect is 0, no action potential. In the second one, we have the example of a spatial summation. We have 2 excitatory potentials from different neurons. They add to each other until the axon hillock reaches threshold, and action potential is produced. And on the right side, we have the temporal summation. Notice that we have a single neuron firing very frequently, and this will produce the same effect. Axon hillock reaches threshold, and an action potential takes place. So this allows us to understand how neurons generate action potentials and allow signals to propagate I'm sorry, okay, to other neurons, to other effectors. And also we understand the importance of the axon hillock, which is the integration center of the neuron, is the beginning of the axon, okay, the region of the axon that is closest or nearest to the soma, okay, where the action potential initiates. It's also called trigger zone, initial segment. This area is the part of the neuron that has the highest density of voltage-gated sodium channels. Okay, in the hillock, then we have many sodium channels, many voltage-gated sodium channels across the axon, but the hillock is the area with the highest density. Then, if you remember, in the axon, there are these nodes of Ranvier that are located in these gaps between the Schwann cells. So these areas also have a very high density of voltage-gated sodium channels, but not as many as in the axon hillock. Okay, the action potential is this transmission of electricity created by the opening and closing of different types of voltage-gated channels. Okay, it can be sodium channels or potassium channels. Okay, these sodium channels, when there is a certain voltage inside the cells, may also be inactivated. Okay, when we inactivate these channels, okay, we are gonna get something that is called refractory period that we are gonna see in a while. And we are gonna see the importance of these refractory periods for the proper control of the nervous system activity and the transmission of signals throughout the body to different effectors. Another important concept is that different neurons have a different speed of propagation of the action potentials. Okay, the speed of propagation, the velocity of the action potentials will depend on a couple of factors. One of them is the axon diameter, and the other is the presence of myelin around the axon. Okay, myelin is important because it is like the structure that produces isolation, okay, in our electrical wires. Okay, this plastic that we have around the electrical wires that permits that the energy doesn't spread, and of course avoids fires in our house. Imagine that we had the wires without any kind of plastic. isolating them, they would get too hot and would produce fires. Okay, larger axons, okay, will have more speed, okay, will transmit to a higher speed faster, okay, because these larger axons will offer less resistance to the current. And of course, myelinated axons will permit a faster transmission because myelin will prevent, okay, the diffusion of the sodium, potassium electrons or atoms. Okay, so we keep the transmission within the axon and it doesn't spread. Now, it is important to know the sequence of events that occurs in the generation, transmission of the action potential, something that I'm sure you have studied several times. I was reading yesterday these comments, okay, that you put on Blackboard, and I know that many of you have master's or bachelor degrees in biology, exercise physiology, and different types of medical sciences, which is great. And I know you have studied this several times. We are now simply trying to focus on the things that are important, okay, for understanding pharmacology, for understanding clinical medicine and pathophysiology, and of course the concepts that we need to retain forever. What is the sequence, okay, in the generation of the action potential? First of all, we need to produce a depolarization of the axon hillock and the different segments, the different nodes of Ranvier, okay, to threshold. Okay, the stimulus that comes from these graded potentials will summate, okay, until threshold is achieved at the level of the axon hillock, okay, making the inner part of the membrane less negative from the resting membrane potential -70 to -55, okay, which is threshold. At this point, we are going to have the activation or opening of the voltage-gated sodium channels. These are channels that respond to voltage and only open if threshold is reached, if this value is reached. That will lead to a rapid depolarization. Many sodium channels will open, and there is going to be a rapid influx of sodium, and the inner part of the membrane will reach a positive value, okay, plus 30 millivolts. These, for example, and it's important when we study this, try to see the application to clinical practice. Okay, many local anesthetics like Docaine, for example, work by inactivating these sodium channels. Okay, so we don't feel pain when we have any kind of procedure, dental or any kind of sutures or any other procedures. This prevents the transmission of these pain signals to the brain. Now, what happens when the cell reaches this electrical value? Okay, at this point, there is a positive feedback that further opens more sodium channels, leading to an overshoot. We are going to see that later in the diagram. Okay, in the overshoot, there is a positive membrane potential. This is the peak of the action potential. And this is the moment when the sodium channels, voltage-gated channels, will get inactivated. Okay, inactivated. It's important when we later analyze all of this together to understand the difference between a closed channel and inactivated channel. That is very important concept in physiology. Okay, so we are going to go to that concept in a little bit. Okay, we have there the inactivation of the sodium channels. That means that it's impossible to trigger a new action potential. And that corresponds to what we call the absolute refractory period. At the same time, we are going to have the activation of the potassium channels. Voltage-gated potassium channels, they open when there is a voltage of plus 30. Potassium channels have a characteristic. They open and close very slowly compared to the sodium channels. Okay, so at this moment, they are going to be opening very slowly. Okay, and this will lead to the repolarization of the cell. That means going from the plus 30 to try to reach the baseline resting membrane potential of minus 70. But since they open and close very slowly, there is a moment when the potential, the voltage inside the cell is going to go below the original resting membrane potential, and we call that hyperpolarization or undershot. Okay, the cell reaches a membrane potential that is more negative than the one at rest. And at this point, only a very, very, very strong stimulus could trigger a new action potential. Because now there is a larger distance between the actual membrane potential of the cell and the threshold. And then how we go back to normal? That is the action of the sodium-potassium pump that reestablishes everything to the original value. This is better understood here. Okay, here we have the resting state. Okay, the membrane is at rest. There is a membrane potential of minus 70. Notice that the voltage-gated sodium channel is closed. Membrane channels, the sodium channels more specifically, are double-gated channels. They have a double gating mechanism. Notice that here at rest, the sodium channel has the outer gate closed, so sodium cannot go in, but the inner gate is open. Okay, the voltage-gated potassium channels have a single gate. In this case, at rest, they are closed, so potassium cannot move out. That's what maintains the difference, the steady state. What happens when we open the sodium channels? Notice that sodium will move in, increasing, okay, the value of this membrane potential from the negative towards the positive. There is a depolarization. Notice that the two gates are open here, so sodium enters. But at this moment, notice that the potassium channels are still closed. That's what permits, okay, the accumulation of positive charges inside the cell is what permits that this depolarizes and reaches the threshold. Okay, if we reach threshold, okay, is when the potassium channels are gonna open. Okay, once we reach this peak, a positive value, okay, this will produce the inactivation of the sodium channels. Compare closed versus inactive. When the sodium channels are inactivated, the inner gate is the one that is closed. The outer gate is open, but still sodium cannot move in because of this inner gating. Okay, they are inactive. There is no way that you can produce a new action potential because the stimuli that open, okay, the sodium channel is not gonna work. Okay, this inner gate doesn't respond to any kind of stimuli. It will be closed while the membrane potential is in the positive value. At this time starts the repolarization. Notice that now the potassium channels are open. They open very slowly. Potassium starts moving out slowly, and we have this repolarization phase. And then we have at the end the after hyperpolarization. Okay, we have the resetting of the sodium channels. Notice that they are closed again like they were at the beginning, but potassium channels are still open. Remember I told you they open and close very slowly. That's why there is a short period of time during which sodium channels are closed, but potassium channels are still open, and that's why the cells reach this even more negative value, which is an absolute refractory period. Sorry, this is the relative refractory period. The absolute refractory period is when the sodium channels are inactivated, okay? When they are inactivated, so from here to here, we have the absolute refractory period. And during this time, until the cell reaches the base, the resting membrane potential again, we have the relative refractory period. Okay, absolute, inactivated, relative, hyperpolarization. And here we have the concept of this speed of propagation, okay? Conduction normally goes faster, okay, when there are these myelin cells, Schwann cells, for example, okay, because in the case of myelinated fibers, all of this process of depolarization, repolarization, etc., will occur here, and then in the next one, and then in the next one, of these nodes of Rambier. While if you have a fiber that doesn't have myelin, unmyelinated fiber, all of this has to be occurring very slowly from one channel to another, so it's going to be very slowly. Okay, here we have a representation and examples of different types of fibers and their conduction velocity. Okay, below you have the fibers, okay, that transmit more slowly. Here we have the velocity, and here we have the diameter. Okay, fibers with a very small diameter will conduct very slowly, while fibers with a large diameter will conduct very fast. Okay, it's not necessary that we now study and try to memorize all of these fibers. Okay, you can rest assured that you can practice medicine without knowing this in too much detail and simply keeping the basic concept of large myelinated transmit faster stimuli, while small diameter unmyelinated transmit very slowly. Okay, now it's important to know examples. Well, yes, let's try to look at the extremes. Let's try to look at the unmyelinated fibers with small diameter. Which fibers are those? The ones that transmit slow burning pain and visceral pain. Difficult to localize pain. Okay, very vague pain, but it's very, very irritating to us. What are the ones that transmit the faster stimuli? Okay, we have, for example, the ones that transmit proprioception, which is the position of our limbs, the position of our joints, the degree of stretching of the tendons and ligaments. Okay, and the fibers that transmit to muscle, skeletal muscle fibers, striated muscle fibers. Okay, these are the fastest skeletal muscle fibers and proprioception. And then we have others in between, okay, others in between, but I just want you to remember the extremes, slowest and the fastest. And the importance of myelin, okay, this is a glial cell, this could be a Schwann cell, for example, in the peripheral nerves. Notice that there are many, many layers of cytoplasm and cell membrane surrounding the axon, like the plastic in an electric wire, so the current doesn't spread, okay, and we can transmit these impulses a lot more faster. Okay, so this is more or less what happens, okay, trying to summarize what happens in the transmission and generation of the action potentials. Okay, we mentioned that after the action potential is generated, okay, and reaches the action end, okay, something is going to happen there. There we are going to have the opening of certain voltage-gated channels, and the release of a neurotransmitter that will bind to dendrites in the following neuron, creating a postsynaptic potential, another receptor potential. The process will begin again, or this can reach a muscle at the level of the neuromuscular junction, doing exactly the same. Okay, let's take a look at this, how neurotransmission occurs. We are going to take the example of the neuromuscular junction, okay, and that will serve, okay, for the next topic that we will have next week, that is muscle contraction. Okay, when the action potential reaches the axonal end, okay, we are going to have another sequence of events that we need to study very well. The neuromuscular junction has different components. Okay, the motor nerve terminus, which is known as the exact presynaptic button or axonal end. And then it has the postsynaptic muscle end plate, which is the one that has the receptors. For example, for acetylcholine, okay, we are going to study later, and you're going to study in more detail in pharmacology, the different receptors that can be there, muscarinic, nicotinic receptors, etc. And also, as a part of this neurotransmission, we have the neurotransmitters, which are the ones that are released from the neuron to the motor end plate, NT, which one example is acetylcholine. Now, what is the sequence of this? Okay, we were talking about the action potential. Okay, imagine that the action potential is reaching the presynaptic button. Okay, that will depolarize the axonal end by opening some voltage-gated calcium channels. And please pay attention to the difference in the channels that we have across in different areas of the neuron. Remember that in the dendrites, we have ligand-gated channels. In the axon, we have voltage-gated sodium and potassium channels. And in the axonal end, we have voltage-gated calcium channels. Okay, this in the dendrite may respond to maybe sodium, maybe chloride, maybe potassium, okay, maybe different things. Now, when these voltage-gated calcium channels open, calcium will enter inside the cells, inside this axonal end, and this change, this increasing concentration of calcium will permit the release of acetylcholine by exocytosis. This is a process that is very complex. First of all, acetylcholine is stored there in certain vesicles. These vesicles first have to bind to the cell membrane. There are certain proteins that participate in this. Okay, after this docking of the vesicles, the membranes will fuse and acetylcholine will be released. There are different things. This gets a lot more complex than this, but we are trying to go to the basics. When acetylcholine is released, it is going to bind to the receptors in this motor end plate. Let's say acetylcholine is here. And this is a type of ligand-gated channel that will open when this acetylcholine binds there. Okay, and these are channels that are channels, for example, for sodium. And when these channels open after acetylcholine binds there, sodium is going to enter inside the cell. Okay, and if there is a good amount of sodium entering inside the cells and the cells reach threshold, that will create an action potential that will lead to the sequence of events that permit the muscle contraction that we are going to be studying next week. Okay, stimulus is going to move through these T tubules, will open voltage-gated calcium channels that are present on the sarcoplasmic reticulum of the cells. Calcium is going to move into the cytoplasm, and actin and myosin will interact and lead to the muscle contraction. Now, we can't forget something. All of this was initiated by acetylcholine binding there. Okay, and we don't want to have muscle contractions that last forever. So we need to do something with this acetylcholine, with this neurotransmitter. In the case of acetylcholine, there is an enzyme in the synaptic cleft that is called acetylcholinesterase. This enzyme will break down acetylcholine into choline and acetate, kind of vinegar. And choline is going to be taken back. The choline part is going to be taken back into this presynaptic button to make more acetylcholine, recycling. How many years have we humans been recycling? Like 2, no? Compared to the amount of time that our body has been recycling things. So that's the process that happens at the neuromuscular junction, okay? Reuptake of choline after acetylcholinesterase breaks down acetylcholine into choline and acetate. Okay, and then we have, don't forget these ligand-gated channels that will let sodium in, initiating another action potential. And that's, in a nutshell, how neurotransmission, or in this case, neuromuscular transmission occurs. But a very similar process happens at the level of the glands and from one neuron to another. Okay, so by studying, well, one of these, we can apply the same concept to other forms of transmission or neurotransmission that we have in the body. This diagram just represents what we were explaining before. Okay, we have here the synaptic vesicles. Okay, we have here the voltage-gated calcium channels. Okay, when the action potential reaches this point, these voltage-gated channels will open, calcium will enter, and calcium here is going to lead to the docking of the vesicles and the release of the neurotransmitters. There you can see the release of the neurotransmitter, the binding to postsynaptic receptors, which are ligand-gated channels, more exactly sodium channels. Sodium will enter in, producing a depolarization of the inner membrane and an action potential. And some of these neurotransmitters are going to be degraded, okay, and taken back into the presynaptic button by a process known as reuptake. Notice that here, imagine that this is choline. Choline is going to be transformed into acetylcholine again, so we can reuse the acetylcholine. Okay. What happens if this choline is damaged? Imagine that the enzyme broke down choline too much and it's not worth it. Well, the damaged choline is going to be taken by this retrograde transport. Retrograde transport to the rough endoplasmic reticulum. Okay, it's going to be broken down into different amino acids. These amino acids are going to be used by the cell to make more acetylcholine that is going to be taken down again by this antegrade transport. And that's the way neurons work. Not that simple. That's a lot of processes there, but we are not doing a PhD in neuron activity. Now we are going to be studying different types of neurotransmitters during our physiology course. Okay, some of them are inhibitory, some of them are excitatory, some of them are both, depending on which is the structure that they are acting on. Okay, there we have the most important inhibitory neurotransmitters, GABA, dopamine, and serotonin. GABA is the most important one. This is the most important of the inhibitory neurotransmitters. Dopamine, for example, inhibits the production of prolactin in the pituitary. Serotonin inhibits anxiety, and we are very anxious. Serotonin calms us down. We're going to have time to talk about that. And here we have the excitatory ones. Okay, acetylcholine is the most important one. Also, glutamate is very important. We're going to see why. Excessive glutamate produces a lot of damage. Okay, to our cells in excess. And then we have epinephrine, norepinephrine, histamine, and also dopamine. Okay, notice that dopamine can be both. If it is acting on one structure, it will inhibit. If it acts on another, it can stimulate that one. Okay, dopamine, for example, makes us feel happy, excited. Dopamine rush. Okay, while serotonin is a great neurotransmitter, but makes us feel good, not excited. It's simply feel good and calm. So the last topic that we are going to touch today is the neuronal circuits. Okay, and we were talking about the synapses. Okay, contact zones between the cells, between cells and effectors. Okay, it's a great structure that permits communication, information transfer, signal transmission, control, regulation, homeostasis. And we mentioned before that can be between two neurons. In this case, they are called neuronal synapses, or between a neuron and a muscle, neuromuscular junction, or between neurons and glands. In that case, they are called neuroglandular. Okay, and they are arranged in many different ways. Okay, if you look inside any electronic device, you are going to see different circuits with different arrangements, and they are very interesting to look at and very difficult to understand, and we don't need that. We simply call someone when the device is broken. And the arrangement of these synapses is what makes us classify the circuits. Okay, in the same way that in electronics you have circuits that are in parallel, in series, etc. Okay, we have several circuits in the body. Okay, they are called neuronal pools or circuits. And this is the classification. Okay, we are going to be seeing the examples of these. One-to-one, one-to-one, one-to-many, so one-to-many, many-to-one, so many-to-one. Okay, and there are also reverberating or oscillatory. So it's like something that reverberates, forming like a circle, and parallel after discharge. That we're going to see, that's better understood with the diagram. Now, let's take a look at these. One-to-one is easy to understand, okay? This simulates that. One-to-many are also called divergent, okay? And here we can give an example, okay? Divergent. Several muscle fibers can be innervated by a single motor neuron. You have a motor neuron that is stimulating 100 muscle fibers. Okay, from one, you stimulate many. One-to-many. Convergent, we can set the example of the respiratory centers. And also the decision that we made to stay in that restaurant. We consider different factors to decide if staying or not in that place. For example, the respiratory centers receive input from the lung stretch receptors, from the chemoreceptors in the aorta and carotid, from the hypothalamus that is measuring the pH of the cerebrospinal fluid, the blood, and the concentration of CO2. Okay, the respiratory center also knows if you are at the gym. Just by standing at the door of the gym, it's going to increase the respiratory rate because somehow it knows what you are going to do. And then we have the reverberating, are circuits that reinforce a response. and create patterns like the respiratory rate. Okay, let's say we are sleeping and everything is fine, and we decide, our brain decides, okay, the respiratory rate is going to be 10 per minute. You don't need to have the respiratory center sending signals so you breathe 10 times per minute. Simply sends a signal that is going to be reinforced by a circuit, and automatically it's going to keep our respiratory rate, unless something happens. We are sleeping and then suddenly there is an explosion. Oh my goodness, that's going to be interrupted and we are going to start breathing faster. Okay, that can be interrupted. Those reverberating circuits explain, for example, some cases of epilepsy and also some thoughts that we may have that repeat themselves and they don't go away. And maybe that song that we say, oh my goodness, I've been the whole day with that song in my mind. Okay. Ideas about depression, about paranoias, and this and that. Okay, that explains many different things that you're going to be learning in the future. Just a diagram to see how convergent circuits are, many to one, divergent, one to many. You say here, you can say muscle fibers. Let's say this is the respiratory center that is measuring oxygen, CO2, okay, it's measuring pH, it's measuring the stretch of the lungs, because you don't want to stretch them too much. They might explode. If you try to inflate your lungs too much, then your brain is going to say stop. Okay, and the decision is taken there. And the last one here, then we have the other one that after discharge. This is the reverberating. Okay, let's say this is the respiratory center that is firing to the muscles of respiration. And from this neuron, you're going to have an axon that is going to stimulate another that is going to recycle the signal. So we don't have to work too much here. The reverberation. And this is an interesting one. Okay, this is the one that explains parallel after discharge. The best example, okay, to this to explain this is what happens when we are blinking. Let's say this is the retina. This is the optic nerve. Retina. There are different neurons there with different axons. Okay, let's say we are looking at a person face and we are blinking. We blink several times. So there is a moment when the image, the light, is interrupted. Okay, but our retina is organized in a way that has a combination of divergent and convergent circuits that are composed by different sets of neurons in their pathway. So in some fibers, the speed of propagation is very fast, not so fast, and slow, because it has to pass through different processes of neurotransmission. Okay, so when we receive these images in our visual cortex, they say this is the cortex, the one that interprets and integrates all these signals, we don't appreciate, or we are not aware of the moment when we are blinking, unless you do it on purpose. Okay, and normally we are not aware, we don't have the interruption, okay, of this image. At least we don't notice it. Okay, and that's what explains also when you are looking, for example, at an image for a very long time, or at something for a very long time without closing your eyes, and then you look at, or you close your eyes, for example, you can see the lights, the shape of that person, or that monitor, or that light, or the sun, if you were looking at the sun. Don't do that, please. Okay, because we still have the image when we, let's say this image got to the retina, to the visual cortex, and we see the person, then while we are blinking, we still have this signal entering, and then this signal entering, and when we open the eyes, the first one goes very fast and we don't miss anything. Okay, that's the best example about that. And this is all for today. And I hope you liked it. And wanted to ask if there are any questions.